Welcome everyone to today's Revive webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Victor Kwasi, and I'll be hosting today's session on Introduction to Antibiotic Research and Development. Revive is GARDP's education and outreach program. It aims to support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All of our webinars are recorded and freely available to view after the live session on our website, revive.gardp.org/webinars. You can also read our series of articles known as Antimicrobial Viewpoints, where experts discuss various topics within the field. And on Revive, we also have a resources section, which includes the Antimicrobial Encyclopedia. This has definitions of various important terms, and some of these will be within the presentations given by the speakers today. And so you can find further explanations here, also with videos where experts explain further the terms. As always, today's presentations will be followed by a Q&A session. You can submit your questions at any time by clicking on the questions button as shown on this slide. We will address the questions after the presentations and do our best to respond to as many as possible. If a question is for a specific speaker, please do indicate their name at the beginning of your presentation. You will have noticed that the GoToWebinar software now has a new interface, so please do let us know if you have any feedback or suggestions in relation to this. There will be a survey after the webinar. Today's speakers are Alan Hennessy, Marianne, and Herbert Wetli. Our moderator is Rosemary Dorrington. Rosemary Dorrington is a professor in marine natural products research at Rhodes University, Makanda, South Africa. She's a South African principal investigator of the SA UK Antimicrobial Drug Discovery Hub and leads a multidisciplinary team engaged in research across the broad field of marine biodiscovery, focusing on the potential of bioactive small molecules as molecular probes for drug development. Welcome, Rosemary. I now hand over to you.
Thank you, Victor, for the introduction, and welcome to everyone who's joining this webinar. I'm going to hand over straight away to our first speaker, who is Alan Hennessy. He is the head of discovery and exploratory research and is the team lead at GARDP. Alan's talk is entitled An Overview of Modern Antimicrobial Discovery. Over to you, Alan.
Okay, thank you very much, Rosemary. Okay, let me try and adjust the slides. Okay, so today I'll be talking about antibiotic research, and it's very much from a medicinal chemistry point of view, okay, and this will be to complement nicely similar research webinars given recently at the Revive Forum. The central premise is that antibiotic R&D has fallen behind. Half of all antibiotics used today were discovered over 60 years ago. Since then, R&D has slowed while drug-resistant infections continue to increase. Today's presentation, I'll give an overview of the discovery process. I'll give examples of the discovery approaches for products, clinical compounds, lead compounds, etc., from a medicinal chemistry point of view. And finally, I'll wrap up with some recent trends in antibacterial discovery. On the right-hand side, you'll see a funnel which describes the process of R&D in categories from the very beginning, screening for new hits, through hit to lead, lead optimization, candidate profiling, and preclinical development. And this is what we would call discovery and exploratory research at GARDP. It's the research phases before a project would enter development, with clinical development ultimately, hopefully, leading to the marketplace. It's shown deliberately as a funnel to indicate the high attrition of projects which fail going through this pipeline. As mentioned, there's been several excellent webinars on similar topics, so I will link them there and advise you to listen to the recordings if you haven't seen them live. Okay, so looking at the stages of early research from basic science to target ID and target assessment, we then move through the stages to hit expansion, hit to lead optimization, ending with an IND filing. The time taken is indicated nominally in years, but this can be highly variable depending on projects. And given at the bottom there are references which describe the attrition rates at every stage of the process. And if I get across one message today is that really we need to increase our efforts in every stage of the antibiotic R&D process, okay? The golden age of antibiotic discovery, well, this was in the middle part of the last century when many, mostly natural products, antibiotic classes were discovered and many semi-synthetic derivatives thereof really dominated new registrations of antibiotics. There were some synthetic chemical classes that I'll also discuss today, but most of those were natural products. Should be said, since the turn of the century, launched antibiotics have really just been additional members of known antibiotic classes, okay? And this lack of innovation really in discovering new classes will hamper our fight against antimicrobial resistance. And indeed, looking at the recently published work from the WHO, looking at antibacterial agents, but the clinical and preclinical pipelines, we see here for the clinical pipeline that there's not many novel mode of action agents in the pipeline. There's some examples of novel topoisomerase inhibitors, FABI inhibitors, for instance, but the pipeline is really dominated by existing classes. And one of those clearly at the top there is the beta-lactam, beta-lactamase inhibitor combination classes. Okay, so on this starting a new research project, you might consider some of the factors you should take into account. Okay, one is described in previous webinars as starting with the desired outcome and thinking about your target product profile. Okay, and several sample profiles are available for certain disease areas from WHO, from Carbex, and so on. So it's important to think about where you want to end up with your product. And also because you can't know at the outset many of the clinical features of a product, an intermediate stage might be assessing what you want in a target candidate profile where you want to end up really at the end of the research phase, okay? And every stage from hit identification, hit to lead, and so on, increasingly builds knowledge across all of the features of your candidate profile from chemistry, microbiology, and toxicology. And again, other resources might help to frame your new project in terms of what's also going on in the field. And there's this excellent report, as I said, from the WHO, which kind of shows the preclinical pipeline in terms of the mechanism of action, whether it's a direct acting or combination agent, which kind of pathogens these projects are addressing and where they're being done. So excellent positioning of your new project would be to understand what's going on out there in the wider field. And indeed, also from the WHO's the bacterial priority pathogens list. So important to address these some of these critical pathogens in your likely research project. Okay, these are pathogens of current importance and likely future importance too. The discovery cascade has been well described as well for all projects, okay, across disease areas, and in specific for antibacterials, specific consideration could be given to resistance to make sure you're screening against the appropriate resistant pathogens from an early stage. Also considering that antibacterials are also dosed usually higher than agents in other disease areas, and they demand a high safety window, okay, so because of the safety of existing agents on the market. Cost of goods is usually kept quite low, so the complexity of your chemistry should be kept in mind, especially if you hope to achieve a global kind of marketplace for your new agent eventually. Some advantages in antibacterial research compared to other areas such as Alzheimer's, for instance, are the translation of activity from early wholesale antibacterial activity will translate quite well often to animal models of infection, which in turn translate well to phase 2 clinical trials, for instance. So that translation is at least a little bit better than in other areas of drug discovery, where this you can have late stage clinical failures due to efficacy. Reasons for failures are quite different often in antibacterials. Okay, so if you're starting from the beginning and looking for screening novel chemical matter to find new chemotypes to start your project, GARDP have published recently on our efforts in screening against multi-drug resistant Acinetobacter baumannii and Klebsiella pneumoniae from the beginning, which is a novel approach and indeed is also to be taken into account the holistic profile of this screening. So early on in the cascade, we will also screen for mammalian cytotoxicity and red blood cell hemolysis, for instance, to avoid disappointment later on. Okay, so getting that kind of early holistic profile is key. Also, there's many opportunities for starting a project building on existing knowledge in the field. Okay, and there's a lot of publications on targets that haven't quite made it yet to the clinic. We would term them unrealized targets in our recent reviews. So projects can start by building on the knowledge already there, or indeed on screening targets that have been recently genetically validated, for instance. So there's lots of discovery opportunities as to where you might start a project. Now I want to change tack a little and look at discovery approaches, okay, and ones that have been successful in recent times and indeed historically. So looking right back into the last century and looking at the marketed antibacterials of synthetic origin, we see that they were mostly originally identified by phenotypic screening. This is also true of the natural products I mentioned as well. So, for instance, we have the story of the sulfonamide antibacterial class, which was originally from Bayer screening of their adjacent dyestuff industries in the 1930s, which led to many sulfonamide products eventually reaching the market. Similarly, naldixic acid was discovered by the screening of chloroquine synthetic byproducts shown in the bottle middle there, and indeed this led to the whole class of fluoroquinolone antibiotics. Okay, so that serendipitous discovery really led to another major class. As for the oxazolidinones, DuPont in the 1970s screened compounds from their adjacent plant science division, discovered this hit, and eventually this led to products such as linezolid and tedezolid, as shown there. So the message here is really these organizations screened every kind of chemical inputs they could get their hands on to find these leads, and then a lot of development from a lot of companies led to the products. So I mentioned that beta-lactamase inhibitors feature strongly in the WHO clinical pipeline analysis. So just looking at the three major classes of beta-lactamase inhibitors and where they came from. So the first is a natural product derived class, beta-lactam class, the original clavulanic acid discovered by Beecham's in the 1970s, which makes an excellent combination with amoxicillin to make the augmentin product. But indeed, there's new entries in this field coming out, so such as this one shown at the top from Orchid. The second class came from biochemical research in the 1970s, and the discovery that boric acid, in fact, and aryl boronic acids could inhibit beta-lactamase enzymes, and indeed on and off research into this area led eventually to several products, and also say this clinical compound shown here from Cupex, which looks a very nice compound. And let me go back one. And the final area was from a different research strategy, which was rational design. So researchers proposed that this 6,5 strained lactam system called BDOs would mimic beta-lactams in their reactive chemistry, and so that was proved and has led to beta-lactamase inhibitors such as avibactam, for instance, and also this direct-acting antibacterial shown there, which has been reported by Entasis. So 3 different hit identification strategies have led to different chemical classes here, and all quite important ones. So the messages would be that multiple hit finding strategies have been successful in the past to deliver products. The organization that starts out and finds the hit is not always the one that develops and delivers to the market the eventual product. And indeed, many products can come from that initial finding. The similarity of the original hit to the product can be variable, but I'd say one thing that is very constant is that each of these projects has involved the design, synthesis, test, and analysis of thousands, at least, of compounds between that hit at the lead and then the products, okay? And this is usually building iterative cycles of research knowledge as these projects have developed through their life cycle. I must say there's high attrition in projects, and I've only shown some successful outcomes here. Okay, but what about screening against novel targets today, okay, and finding leads with novel mechanisms of action to address resistance? So the compound there on the top is zoliflodacin, for which GARDP recently reported successful phase 3 clinical trials. But the original hit came from a phenotypic screening, again, from Pharmacia, of their extensive compound library to give this hit compound there, this nitroaromatic, which was much optimized to deliver zoliflodacin, originally disclosed from AstraZeneca. Similar story for this biotin carboxylase project, where Pfizer screened their enormous chemical library against efflux pump deficient E. coli strain and found this hit. And this project was taken on then by Achaogen, who reported a very nice lead compound in 2018, and hopefully this area can be further taken on by other organizations. Another story as well in LPXH is where researchers from University of Uppsala just this year reported on taking hits from both AstraZeneca publications and published screening data, and made a very nice medchem story of merging hits to form a very nice lead product as shown there. What about target-based screening, okay, rather than phenotypic screening on whole cells, looking at the more pathways dependent or in fact target-based approaches? And in this case, we see the discovery of LPXC inhibitors started with the story from Merck, where they had a pathway screen in Salmonella measuring LPS synthesis. And they had these compounds from their collection, these hydroxamic acids, which came from, in fact, their efforts in cycloserine synthesis, found this hit, and elucidated the mode of action as LPXC. And this spawned a huge amount of effort on this mode of action. And unfortunately, several of the leading early compounds failed for toxicology reasons in the clinic. But now there are a revival there, and there's this compound from Duke and also other non-hydroxamic acid compounds in clinical trials. So we wish them all the best with pushing those areas forward. Target-based screening on isolated proteins has been somewhat less successful in the past, as reported by GSK in their high throughput screening efforts. But one success was on FabI, where they reported that inhibitor there in the bottom right. And indeed, this area was taken on by organizations like Debiopharm, who have clinical candidates in this area, and also the University of Illinois. And the compound shown there is called fabimycin, which indeed has much more, say, gram-negative activity than the original hit. Another target-based hit-finding approach is fragment-based screening, and indeed AstraZeneca reported the successful fragment-based screen against the GARDP target to give this little pyrrole ester, which they combined with institutional knowledge to form this clinical candidate shown there, which unfortunately dropped out early in clinical trials in the last decade. Now, as I said, in the golden age of antibacterials, many agents came from natural product screening. And today, it's also the case that natural product screening is important in finding hits. It's just that you have to be doing this process somewhat differently, I think, to how it was done before. So to find new bugs and screen them in different ways, to find new chemical matter with novel modes of action. So I showed there novel acetyl-CoA carboxylase lead compounds from Bayer discovered in this way. Also the arylomycins, these interesting bisphenol macrocycles were very interesting, taken on by Genentech to form interesting leads and maybe clinical candidates as well against the LEP-B target. Indeed, darabactin was reported as a natural product from an unusual source, which inhibits the BAM-A protein, and indeed several researchers are now developing more potent analogues of that natural product. Okay, so in take-home messages, there's again similar hit-finding strategies that delivered historical products, phenotypic screening, but also now target-based screening and advanced natural product screening. So all of these areas have benefited from technological increases. And it's also should be said that we need to accelerate and redouble those kind of efforts to refill the pipeline, which, as I said, somewhat lacks in innovative products at the moment. Can we do it better than in the past and make this more efficient with recent technology discoveries? And I'll just highlight a few examples here where I think the answer is yes. So certainly one major advances in physical property understanding and work from Paul Hagenrother at the University of Illinois was quite transformational in formalizing the understanding of the physical properties needed for agents to get into gram-negative bacteria, and really providing great design inspiration for development of broad-spectrum antibiotics. Second area is looking at chemical inputs. So chemical inputs from the last century, wherever companies could find them, and often in adjacent industries, as I mentioned. But now the available commercial chemical input universe is growing exponentially, with the likes of, say, enamine real, for instance, offering a lot of chemistry to screen. But also diverse chemistry can be found in unusual places, such as these unconventional small molecule macrocycles, as shown by Roche in their discovery of zosurobalpin. So they reported this earlier in the year, and a great story on both the chemical input, the optimization of the hit to the clinical candidate, but elucidating mode of action, overcoming toxicology, shows really a excellent integrated drug discovery process. Also AI, beyond the hype, I think we see that AI can have a real impact in the future in antibiotic research. And we see already discovery of compounds which have, and not just theoretical interest, but real in vivo activity. Okay, so some excellent papers to refer to there. Also non-traditional approaches. Okay, so I only really showed the classic direct-acting antibacterial small molecule area, but non-traditionals are growing and have been really well covered in a recent Revive webinar. I'd direct you to that. So in conclusion, there's a real need for innovative antibacterial products everywhere in the pipeline, okay, from hits to leads to candidates and on into the preclinical pipeline and onto the market, okay. But I think recent areas have shown that, the innovation that has increased in recent years offers us hope there, and this process just needs to be accelerated, and indeed later assets, when discovered, need to be carefully nurtured to the market, okay. And okay, that's the end for me, but thank you very much for listening, and also thank you to my GARDP Discovery colleagues in helping to prepare these slides today. Okay, thank you.
Thank you, Alan, for your comprehensive review. I would just encourage participants in the webinar, please to post, if you have any questions, please post them in the question section, and we will have a question and answer session for all three speakers at the end of the webinar. Thank you very much. Our next speaker is Mo Yin. Mo is an infectious diseases physician and a clinical scientist based at the National University Hospital in Singapore. The title of her talk is An Overview of Antibiotic Clinical Trials. Mo, over to you. Thank you.
Thanks, Rosemary. A very good day to all. I'll be sharing with you today my insights on phase 1 to 3 trials for antibiotic development. I hope to offer a top-level understanding of the pipeline challenges and complexities of the development of new antibiotics from the point of view of a clinical trialist. Here are my conflicts of interest. So in the next 15 minutes or so, I'll be covering these two major topics. I'll start off with an introduction to what are phase 1, 2, and 3 trials for novel antibiotics. I'll provide some real-world examples to illustrate antibiotic-specific challenges and explain some methodological considerations in trial design and also regulatory approval and ethical issues. We'll first start off with some basic introduction to phase 1 to 3 trials for novel antibiotics. So for an antibiotic to enter the market, it starts off with antibiotic discovery and preclinical studies, as Alan has highlighted earlier. About half of these eventually enter human trials, starting with phase 1. And these trials are first in human and usually involving a small group of healthy patients. These are mainly focusing on finding the right dose and ensuring safety. Subsequent phase 2 and 3 trials are increasingly seeking signals of efficacy while continuing to collect safety data. Sample sizes also become increasingly larger. As you can see on the top graph over here, few candidates from drug discovery stage actually end up in the market. So this is a typical phase 1 trial setup as shown in the picture on the right upper hand corner. This involves usually small groups of patients. The design may be randomized or not and typically open label. Healthy volunteers with normal organ functions are enrolled in order to study pharmacodynamics, which refers to biological and physiological effects in humans and its mechanisms of action. Pharmacokinetics are also studied, which include the drug's half-life, plasma concentrations in various organs, and bodily compartments. The design is typically a dose escalation design where participants are given increasing doses until a maximum tolerated dose is identified. And if possible, phase 1 trials may also provide early signals of efficacy, especially via surrogate markers of clinical or microbiological success. This is an example This is an example of a phase 1 trial targeting Staphylococcus aureus. It involves an antibiotic antibody conjugate. You may appreciate that this trial is a randomized, placebo-controlled, single ascending dose trial involving a small group of healthy volunteers. And the main focus is on safety and pharmacokinetics. Early efficacy signals is detected via immunogenicity. And moving on to phase 2 trials. Usually, these type of trials involve diseased patients and larger sample sizes. Sometimes this may be split into phase 2A and 2B trials, and these labels are largely arbitrary and usually communicates a shift from safety and PK/PD to efficacy as the main focus. In phase 2A trials, these are usually used to establish preliminary dose-response relationships. It explores efficacy using surrogate or exploratory endpoints, and at this stage, the dose may still be varied. In phase 2B trials, the methodology is usually more definitive in the endpoints, and such trials will need to be carefully discussed with regulatory bodies to align expectations in terms of making a go or no-go decision for subsequent phase 3 trials. And this is an example. And this is an example of sorry about the slides. Okay, this is an example of a phase 2 trial. It's the APEX complicated urinary tract infection trial for cefiderocol. You can see that this involves many countries and hospitals enrolling over 400 patients. The primary endpoint is similar to a typical phase 3 trial, which included composite clinical and microbiological outcomes at test of cure. And finally, moving on to phase 3 trials. These involve usually diseased patients and typically of large sample sizes and multi-centered. The methodology is the most rigorous and has to be aligned with regulatory body requirements. Blinding is ideal, however, this may not be feasible, especially when the comparators are made up of a mix of different best of care antibiotics, and the clinical endpoints are typically definitive, such as mortality and clinical cure. An example landmark trial which granted the FDA approval for sulbactam durlobactam is the ATTACK trial. This was done in 16 countries and almost 60 hospitals. The primary endpoint was mortality, since it was targeting patients with severe infections such as pneumonia, bloodstream infections instead of urinary tract infections. And this is a non-inferiority trial, which is the typical design for novel antibiotic trials. Even though the number of patients enrolled was quite small at about 180 patients, and the non-inferiority margin was fairly large at 20%, the drug received FDA approval. Okay, and this is in contrast to tebipenem, which is a new oral carbapenem. And this phase 3 trial was conducted on patients with complicated urinary tract infections, and the comparator was ertapenem. And this was based on over 1,300 patients. The trial successfully showed non-inferiority in terms of clinical cure and microbiological response. However, the FDA did not approve the drug, citing the main reason as insufficient data. Another phase 3 trial is being conducted at the moment to hopefully fill this gap. And the third example is cefepime-taniborbactam. This is one of the rare antibiotics in which its phase 3 trial actually showed superiority compared to meropenem as a control arm amongst patients with complicated urinary tract infections. This is highly unusual as the typical methodology is non-inferiority, which I'll explain later on. However, again, cefepime-taniborbactam failed to receive FDA approval despite not having any clinical safety or efficacy issues. And the main concerns here was over chemistry, manufacturing, and controls issues, which will be discussed in detail by Herbert in the subsequent talk. So that concludes my first section of the talk. We went through the key principles for phase 1 to 3 trials for novel antibiotics and introduced some challenges through real-world examples. In the subsequent section, we'll go in depth into some of these challenges specific to antibiotics. So the first issue we cannot avoid mentioning is profit. As you can see here in this very comprehensive study commissioned by the US Department of Health and Human Services, the median development and approval costs, including post-approval phase 4 studies for antimicrobial drugs, were nearly the same as oncology drugs at about 150 million US dollars. Let's zoom into the antibiotics. Okay, the number of patients enrolled for antibiotic trials was similar to oncology drugs, which is the main driver for costs. The average number of patients enrolled for the clinical stage, which included phase 1, 2, and 3 combined for antibiotic drugs, was 2,400, which was comparable for those in the oncology cohort. However, when we look at the returns, the cost of developing an antibiotic is 2 times less than an oncology drug, but the returns are 10 times less than an oncology drug in the first year. If we can look at the y-axis on these graphs, the peak performing antibiotic in terms of quarterly sales is just at 100 million compared to 1,600 million for oncology drug and 350 million for a non-antibiotic, non-oncology drug. So just looking at the profit margin for novel antibiotic development is important, but it's also a narrow approach considering the potential societal benefit of novel antibiotics. In the Graham study published in Lancet based on pre-COVID 2019 data, AMR was already the third cause of death in global disease burden, surpassing HIV and malaria. Despite the high costs and risks associated with novel antibiotic development, a variety of push incentives such as R&D grants through governmental and other public-private partnerships has lowered the estimated development and approval costs for select antimicrobial drugs, making them significantly lower than those drugs in the non-antibiotic drugs and oncology drugs. In fact, in a recent review published this year, there were 50 novel antibiotic candidates in early phase trials, i.e. 1 and 2 trials. However, downstream challenges persist for these novel antibiotics to reach market eventually. And let's go through these challenges in detail. So a major issue in enrolling patients into clinical trials, especially phase 2 and 3 trials, is because when we think about critical infections such as bacterial infections, the pathology is very complex, considering antimicrobial resistance and the bacterial pathogens themselves. Unlike in most diseases where the host is the main host, when the problem is the main host that we want to consider, here we have 3 axes of consideration. So firstly, novel antibiotics tend to target certain bacterial species and certain resistant mechanisms only, as indicated by the green cells here in this table. Also, there often is no standard of care for comparison in the control arm. So when we look at antibiotic resistance mechanisms, they are fairly complex. Let's take, for example, carbapenem resistance. This can be attributed to carbapenemase production, which refers to an enzyme that can actively break down carbapenems, which is a last-line antibiotic. And carbapenem resistance can also be due to a combination of other resistance mechanisms, such as extended spectrum beta-lactamases, overexpressions of MC, loss of purine channels, and efflux pumps. Each of these resistance mechanisms are preferentially present in different organisms. And next is the host axis. Okay, so the same bacteria may infect different organ systems such as the bloodstream, lungs, abdomen. Each may be associated with its own PK/PD profile. And in addition, patients with multi-drug resistant infections tend to be very ill with major organ failures and sometimes with drug allergies. And this further limits the number of eligible patients for trials. And thirdly, bacterial pathogens Bacterial pathogens themselves may further evolve resistance while on treatment due to the short-term effects of antibiotics and within host selection for resistance genes or increasing propensity for resistance to transmit between hosts. The longer-term side effects of antibiotics, such as de novo mutations or further transmission of resistance to the community setting, are very difficult to quantify in trials. In terms of identifying patients for trials, it may be difficult to define infections when bacterial pathogens may be innocent bystanding colonizers and not true pathogens. An example being ventilator-associated pneumonia, where multi-drug resistant bacteria may colonize the endotracheal tube without causing any infections. And lastly, there are major system issues which prevent clinical trials from being conducted for antimicrobial resistance. In general, there's a mismatch between disease burden and clinical trial capacity. The Global South, which carries the majority of the overall burden for antimicrobial resistance, tend to have low capacity for trials. These countries often lack antibiotic stewardship and infection prevention and control measures to reduce antibiotic-resistant infections. And this raises ethical questions on whether novel antibiotic trials should be done in these places where infection prevention and control cannot be optimized in the first place. In addition, when diagnostics are not optimized, antibiotic trials cannot be done as the efficacy of antibiotics are best captured when they are administered within the first few days of infection onset. There's also a lack of surveillance data to inform disease burdens for study site selections. And from the general public's point of view, there's a widespread mistrust and skepticism for clinical research. So Advanced ID, which I'm co-leading, that's based at National University of Singapore, is currently looking at some of these issues by setting up a clinical trial network. So we now have over 100 hospitals, and we maintain a warm network by conducting a large AMR surveillance study called ACON-HAI. And that study is enrolling at about 120 to 150 patients per week. The main goal of the network is to embed best research practice in antimicrobial resistance hotspot areas to conduct effective and efficient interventional trials through training, capacity building, and providing lab equipment. We also do extensive work on regulatory science, community engagement, and novel trial design methods to tackle the issues I described earlier. Another issue I would like to highlight is the common use of non-inferiority hypothesis in novel antibiotic phase 3 trials. They are widely used because placebo-controlled trials are no longer considered ethical, as existing antibiotics are highly efficacious for antibiotic-susceptible infections. In non-inferiority trials, we decide on a margin which we are willing to sacrifice in terms of clinical efficacy for its potential benefit in reduction in side effects, costs, and etc. However, this margin is potentially subjective and often require a panel consensus to agree upon. And this idea of accepting not too bad treatment as the new standard of care may pose ethical issues. It is important to note when trials are poorly performed, the tendency that it is easier to show non-inferiority in contrast to superiority. And this graph here illustrates the potential for biocreep, where a series of trials with non-adherence to allocated treatment is being conducted. Where there is poor adherence to allocated treatment, the new treatment may be deemed as not too bad compared to the standard of care. And when the new treatment is accepted as the new standard of care and subsequently compared to the next new treatment, this may create a slippery slope where eventually the standard of care is in fact much worse than the first standard of care when we started off with. So how do we shift to superiority trials instead of depending on non-inferiority trials? And here are some potential solutions. So first, we can consider targeting patients with extensively resistant infections, such as carbapenem-resistant gram-negative bacterial infections, where standard of care, for example, in Asia remains to be polymyxins. And polymyxins are older antibiotics of relatively poor efficacy, which novel antibiotics can be compared to. We can also employ novel designs in terms of randomization, outcome measurements, and analysis to rank treatments instead of comparing pairs of treatments based on arbitrary p-values. and categorical outcomes may be used to detect superiority instead of non-inferiority based on a single binary outcome of mortality. And lastly, when a new antibiotic is combined with with old antibiotic as the intervention, the control treatment is the old antibiotic. Such trials should be of superiority in design. So in conclusion, we've discussed that despite market failures, novel antibiotics are being developed with sorry about the slides again. Okay, are being developed with joint incentivization from governments and public private corporations. These antibiotics are largely in phase 1 and phase 2 spaces, while funding for late stage trials like phase 3, phase 4 trials are still lacking. So despite the high global disease burden, antibiotic profit margin remains low because largely the burdens are in the global south, where antibiotic stewardship, infection prevention control policies are lacking. And these issues perpetuates a vicious cycle of poor access to novel antibiotics, limiting profit margin. So as my last slide, I would just like to highlight this article that's published in Lancet very recently, discussing the discrepancy between strategic priorities in the global north and global south when tackling antimicrobial resistance. In this webinar, we discussed extensively on antibiotic pipelines. However, while novel antibiotics and diagnostics are very important, without good infection prevention and control, antibiotic stewardship, clean water and sanitation will be very challenging to catch up with the issue of antimicrobial resistance for everybody. So that is my last slide. Thank you very much for your attention.
Thank you very much for a thought-provoking talk, Mo. I'm sure that there will be lots of questions for you at the end of this webinar. Thank you very much.
Thank you.
Our third speaker is Herbert Wetli. He is the head of pharmaceutical development at GARDP, and he's responsible for the assessment of new drug product opportunities at GARDP, as well as the coordination of chemistry, manufacturing and controls. The title of his talk is Chemistry, Manufacturing and Controls, CMC, in Drug Development. Thank you, Herbert.
Thank you, Rosemary, for this kind introduction. Good morning, good afternoon, and good evening, everyone. My name is Herbert Wetli, and I'm the head of pharmaceutical development at GARDP in Geneva. And in this presentation, I would like to give you a short overview on chemistry, manufacturing, and control activities related to the development and the commercialization of pharmaceutical products. Now, first of all, what is CMC? CMC is an abbreviation for chemistry, manufacturing, and controls, and it stands for several integrated functions and deliverables that are all related to the development and commercialization of pharmaceutical products. They are equally required to deliver an acceptable product to patients, and none is less important than the other. It includes the drug substance on the far left on the slide, also called the active pharmaceutical ingredient, or abbreviated the API. Further, it also includes the drug product, for example, a tablet, a capsule, or an injection. CMC is also about the analytical methods, both for performing tests during manufacture, but also for clinical or commercial release of the pharmaceutical product. It is also related to the regulatory filing of a pharmaceutical product and related updates. A pharmaceutical product must also follow GMP regulations, which are embedded within quality. And CMC is also about the manufacturing of the API and the drug product at different batch sizes across the whole life cycle, from early development to commercial manufacture. And finally, CMC is also about packaging. The primary packaging is the packaging that is in direct contact with the pharmaceutical product, whereas the secondary packaging is, for example, the carton box that you can see on display at pharmacies, for example. Okay. CMC activities play an important role across the whole drug development path that can be roughly split into 4 phases. The late discovery stage on the left, which involves the selection of the most promising drug substance, followed by the early development stage, which involves first clinical trials, Next comes the late development stage, yielding the filing and approval of the pharmaceutical product. And finally comes the commercial stage on the right, shown here as the marketing stage, which involves life cycle activities of the pharmaceutical product. Okay, so let's now focus a bit more in detail on the late discovery and early development stage. Let's start with the drug substance, or also called the active pharmaceutical ingredient. That is the ingredient that is responsible for a pharmacologic effect. Now, after an API has been identified based on its pharmacologic activity, it does not end there. Most APIs are present as crystals and salts, for example, as a sodium salt crystal or as a hydrochloride salt crystal, and each salt crystal has different properties, as you can see on the right. The salt and the crystal form impact the physicochemical properties of the API at several levels. For instance, the bioavailability, the developability, and also the stability. It is therefore very important to select the appropriate salt and crystal form at the beginning of a development. Now, it's obvious that without the availability of a drug substance, the development cannot start. You need drug substance in sufficient amounts to develop your formulation, for example, a tablet or an injectable. Therefore, the API synthesis is not the critical path of the whole development timeline, and an early development synthesis can easily take up to 12 months or even more, depending on the complexity. Normally, you start with smaller lab-scale batches, which are not intended for clinical use, but rather for characterization tests, and then you gradually upscale the synthesis process to larger batch sizes that can be used in clinical trials. At this stage already, it is important to define a synthesis process that can be upscaled to manufacture larger amounts at a later stage. Analytical methods are also required, both for analysis and in-process testing of the drug substance and the drug product manufacture, but also to prepare a precise drug substance specification. In this way, you are able to identify deviating drug substance batches that have to be discarded or reprocessed, if possible, before using them for drug product development and manufacturing activities. It is important to mention that also the packaging, or in other words, the contained closure system, is a part of the specification because the type of a packaging can have a determinant influence on the product. For example, if the drug substance is light or moisture sensitive. Then you need to select the appropriate packaging, obviously. And finally, let's not forget the drug product in early development as well. The appropriate formulation is key because depending on the formulation that has been selected, various characteristics such as the bioavailability, the dissolution, and the stability are influenced by it. Formulation development goes together with drug substance development, and it might happen that certain salt forms or crystal forms of the API are not compatible with a specific formulation or manufacturing process. These types of interactions need to be identified and clarified very early on, and it is not wise to rush doing these activities. Otherwise, it could happen that you are forced to go back again at a later stage and have to redo some of these activities, leading to development delays and cost increases. Now, all activities related to development activities, manufacturing process performance, analytical performance, and proper documentation are governed by quality guidelines. For this purpose, during the performance of all these activities, a quality management system, abbreviated QMS, needs to be put in place to have a reliable and trackable process during development, manufacturing, and also even during discontinuation. I assume that most of you have already come across the term GMP, which stands for Good Manufacturing Practice, and is a system that ensures that manufactured products are consistently produced and controlled according to set quality standards. We don't have the time in this seminar to go into further details of GMP, but in a nutshell, it can also be summarized in the phrase that anything that has not been recorded did not happen. The ICH guideline Q10 describes a quality system, and it is strongly recommended to use this document as a reference for pharmaceutical product developments targeting global launches. Next, I'd like to share a few words about beta-lactams. Many antibiotics, but not all, contain a so-called beta-lactam moiety in its structure, as shown in red on the picture on the right. It is well known that molecules with such a structure can lead to very severe allergic reactions. The technologies to manufacture tablets or injectables do not differ from medicinal products without beta-lactams or antibiotics in general. However, due to the allergy potential, it is strongly recommended, if not even required by health authorities, that drug substances and drug products belonging to or containing beta-lactams are manufactured in segregated areas in order to avoid a cross-contamination to other products. This means separated air and ventilation systems, gowning rooms, material and personnel flows, and manufacturing areas. In the best case, you just have a dedicated, separated building, and last but not least, a license from the local government is required to be able to handle this class of medicines. A very important tool to frame developments is the so-called target product profile, or more technically speaking, the quality target product profile. It defines very clearly what the target product has to be or not to be. This allows a targeted and focused product development so everyone is on the same page at all stages. Below you can see an example of a QTPP, which defines the administration route, the treatment regimen, the packaging, and the shelf life, for example. For such a purpose, an essential or minimum target is always defined along with a desirable or ideal target. The below example is of course not exhaustive. The target product profiles for other medicinal products, such as injections or topical preparations, are of course different. Okay, now when you start a development, there are always two important things to consider: risks and costs, and you need to decide between the two. Let me say it very clearly: there is no safe, cheap, and predictable development. Development activities have to be tracked, monitored, and continuously evaluated for risks and associated costs. For example, should you wait for positive clinical trial results before preparing the next upscale phase? Or should you already spend money to start improving the manufacturing process as long as stability data is not available? One can certainly postpone important decisions like these because of the associated costs, but you will lose precious time if you wait too long. And if you wait too long, on the other hand, the patent window gets smaller and the exclusivity window before generics start entering the market gets smaller as well. This has to be carefully assessed and balanced against each other. Now let's go over to the late development stage and the life cycle management stage. Okay, so now you have successfully completed the first clinical trials, you have passed clinical phase 1, phase 2A, and you have reached the proof of concept milestone. At this stage, the probability of clinical and regulatory success increases and the development activities shift from development to commercial readiness. For instance, in clinical phase 3, the pharmaceutical product has to be the same as intended for launch later. It cannot be different. At this stage, the drug substance synthesis process has to be optimized for the commercial launch. The process needs to be finalized. It has to be safe, robust, cost-efficient, and ideally also sustainable. The lead time here, depending on the complexity, can take up to 3 years. The commercial manufacturing supply chain needs to be set up, and you need to start transferring the synthesis process to the foreseen commercial manufacturing site. Ideally, if you can afford it, you should have a main supplier and a second supplier in case of issues with the first source. The focus is now on filing readiness, and you need to start generating key data for the submission, such as batch data, stability data, and process validation data, typically on 3 different batches. Now, I mentioned sustainability on the previous slide. Sustainability aspects are becoming increasingly important, and they offer environmental but also economic benefits. By following the 12 principles of green chemistry, by optimizing the yield, and by applying continuous manufacturing approaches, you not only do save costs, but at the same time, you can also reduce the burden on the environment. We are all well aware that antimicrobial resistance to antibiotics is increasing worldwide, And one very effective preventive way to reduce this is by pre-treating waste streams emerging from antibiotic production. Just very recently, the World Health Organization has issued a guidance on wastewater and solid waste management for manufacturing of antibiotics. It contains a lot of useful information on how to reduce the pollution of the environment by antibiotic waste streams. So we have touched the drug substance. Let's now cover the drug product at this late pre-commercial stage as well. Similarly to the drug substance, the focus at this stage is to determine the final commercializable pharmaceutical product. The commercial manufacturing sites have to be identified in case you perform a transfer from a development site to a manufacturing site. Similarly to the drug substance, it is recommended to have 2 different qualified suppliers in order to have a backup in case one site has delivery or manufacturing issues. At this stage, you have to generate the relevant stability data with fully validated analytical methods and the process validation data for regulatory filing. And in case you develop a generic, you have to demonstrate that your product is bioequivalent to the originator product. Now, obviously, not every company has internal manufacturing capabilities, and you are forced to work with contract development and manufacturing organizations, which are abbreviated as CDMOs. They will develop and manufacture the pharmaceutical product, the analytical methods, and perform the stability studies on your behalf. One must be aware that every CDMO has different strengths and weaknesses. They have different types of equipment, and they are localized in different geographies. Perhaps there's a CDMO close to your office, but it lacks the capability to perform the development. On the other hand, you have identified a CDMO that has the required equipment, but it is located in a different continent, and you cannot visit it regularly due to the distance. For this purpose, it is very important that you have quality, technical, and development agreements in place that define the roles and responsibilities, the expectations, and the deliverables. The decision whether to manufacture in-house or to outsource has to be carefully thought through. If you outsource activities, you might certainly save fixed costs, but at the same time you might lose the ability to track and prioritize because you have become dependent. Also, if you outsource, detailed product or process-related know-how is not in your own organization, and this knowledge might be lost if employees working at the CDMO decide to leave at some point in time. And very important to note is that the clinical trial sponsor or the marketing authorization holder is accountable for the quality and safety of the clinical trial medication or the final product. You can delegate manufacturing responsibilities, you can delegate analytical responsibilities, and you can even delegate formal release responsibilities, but you cannot delegate accountability. This is very important. Okay, now you have launched the product in your markets. However, it doesn't end there. There might be additional launches in other countries as well. There could be line extensions. For example, you might want or need to develop a sustained release formulation. You might want to increase the batch size, or you might want to make the manufacturing process more efficient or robust. Or it could even happen that you may have to change the manufacturing organization due to contractual or cost reasons. All of this requires resources in terms of personnel and costs and must be carefully planned, managed, and tracked. So, summarizing what are now the key takeaways. Development and future manufacture is always an at-risk investment and requires careful planning and risk management. In this way, CMC activities do not influence adversely the whole project, especially if they are on a critical path, which they mostly are. Every CMC decision, whether it's the choice of the formulation or the API, must be considered very carefully in order to anticipate and mitigate potential issues later on. You don't want to face serious issues when you file for marketing authorization. Then it's too late, obviously. As mentioned earlier, in developments and in life cycle improvements, it's always a careful trade-off between time, resources, costs, risks, and quality. You might be tempted to shorten the timeline and in this way reduce the costs, but it could have an adverse impact on product quality. Every case and every product is different, and this must be carefully assessed for each situation individually. As highlighted, the goal in early development is to decide as early as possible on the viability or non-viability of a new pharmaceutical product, whereas in the late development stage, after phase 2A, the focus is on optimization and commercial readiness. And lastly, investing on quality is indispensable. If you want to deliver to your patients a stable, effective, and safe pharmaceutical product, both for you as a sponsor or license holder, but also for your partners. And with this, I thank you all for your attention. I hope I could explain you the fundamentals and the importance of CMC. I look forward to your questions. Thank you.
Thank you very much for your informative talk. We're going to move across now to questions and answers. If I could ask all three speakers to, there we go. For those who are attending the webinar, please can I encourage you to post your questions in the question section. I'm going to kick off with a couple, and then hopefully that will set up a lively discussion. My first question is for Alan. And the question, you gave a really good review of antibiotics, the antibiotics in the market, and an idea of some of the new ones. Question for you, do we need to find new chemical entities or novel compounds to continue the discovery of antibiotics? And where do you think we would find those?
Yeah, thanks, Rosemary. Yeah, I think definitely. I hope the all the talks today conveyed the need to repopulate the antibacterial pipeline at every stage, okay, from the earliest parts of discovery, so identifying novel protein targets, to hit identification, so chemically validating those targets with hits, developing those into leads, then passing them into the stages that Mo and Herbert described in development. Okay, so yeah, without a doubt, it seems that from all the recent analyses in the literature that the AMOR problem is probably outpacing the discovery and development of solutions in this area. So yeah, without a doubt, I think for the long term, every part of the pipeline needs to be regenerated.
A question for Mo. You've talked about the problem of not having the clinical trial capacity in the regions in the global south where there is the highest burden of antimicrobial resistance, and therefore the need to be running clinical trials. Do you have some idea as to how this lack of capacity can be addressed?
Thanks, Rosemary, for the question. So this is exactly what Advanced ID is trying to tackle. So Advanced ID is a clinical trial network that I'm co-leading with Professor David Patterson. We are based at the National University of Singapore, the School of Public Health. So over the past 2.5 years, we've been trying to build this capacity in the Asian region, and now we've got over 100 hospitals. So what we do with these study sites are really engaging with the ground investigators and helping them to build up research teams and getting them the right equipment and undergoing all sorts of training from microbiology labs to good clinical practice, GCP, data collection, and sometimes even ensuring there's stable electricity or internet. So we started off with a really big surveillance study looking at both susceptible and resistant severe bacterial infection. And so that's the study called ACON-HAI. And hopefully when the data is ready, it can really highlight sort of individual-based data, the disease burden on patient mortality and morbidity. So that's our first step. And through doing such a robust surveillance study and the capacity we are building, we are moving on to diagnostic trials, new antibiotic trials, existing antibiotic trials. So it's a multi-year journey, but we are very fortunate and extremely lucky to have a group of very, very motivated investigators working together here.
One kind of forgets that the diagnostic trials are really important, because without the one you can't really do the trials for the compounds. Yes, right. So the next question I have is for Herbert, and that is You talk about you've highlighted the need for CDMOs that can be located in the geographical location of where these compounds are needed. How does one ensure that the quality of these entities is maintained throughout the process?
Thank you, Rosemary, for this question. No, indeed, that's a very important question. Obviously, many CDMOs are spread all over the world. They can be in Asia, they can be in Europe, they can be even in America, in the US. One very good point to start is, for example, to go to the site personally, perform a GMP audit, a good manufacturing practice audit, also to perform a quality audit to see whether they have a qualified system, a quality management system in place to track deviations, failures, change controls, etc. Last but not least, it's also very good to meet the technical people face to face, not just the marketing people or just have a look at the website, also to have a personal impression on the site. Normally in our projects, we go to the sites at least twice per year and have a technical discussion also. We make a tour around the labs to get really a good feel of what they are doing. And lastly, as I mentioned in my presentation, it's very important to have very well-defined agreements. I mentioned quality agreements, technical agreements, also development agreements and master service agreements, but define very clearly the responsibilities, the delays, and the deliverables and the expectations. But of course, I mean, given to the distance, it's a bit of a challenge. It would be better to have the CDMOs next to you. And besides that, what we do, we have a bi-weekly calls. So every month, 2 calls per week with the companies, with the organizations, and to track the progress. Thank you.
A question for the whole panel, and I would be interested in your comments. How do you see the post-COVID environment affecting the field of antimicrobial drug discovery? And have certain parts of it improved? One would think perhaps the capacity for clinical trials and manufacturing potentially, or is it just exposed the need that has already, the underlying need that's been there all along? Perhaps we start with Mo.
Oh, okay.
Thanks, Rosemary.
On my screen.
Yeah, I can share the perspective from clinical trials. So there's been a lot of positive changes, I would say. Firstly, there's sort of an agreement amongst a lot of big funders and WHO and international health bodies and authorities that there's a big gap in clinical trial networks. It is especially in the setting of pandemics, it takes too long and too much of an effort to put together trial sites, having to standardize protocol and meeting a minimum clinical trial standard when pandemic happens. So there's this idea of a peacetime clinical trial network idea, and that's potentially focusing on endemic infectious diseases like dengue or AMR that can quickly pivot when the next pandemic occurs. So that's the first positive change. The second thing is that there's been an increased, a lot of interest in platform trials. So platform trials are these ongoing studies that test multiple different interventions, and they're usually Bayesian adaptive trials that will allow new interventions to be added as they become available or drop existing trial arms if they are deemed to be not meeting certain expectations during interim analysis. And that is very useful for antimicrobial resistance as well, because there are multiple existing treatments for the same pathogen, same resistance mechanism. So these are the two positive changes. And one negative change I would say is because during the pandemic, there were a lot of antibiotics being used on COVID patients. And in general, we see a uprising trend, especially in gram-negatives and carbapenem-resistant gram-negative infections. It's highly prevalent, especially in Asia. Yeah.
I'm going in a clockwise direction. Herbert, would you like to comment?
Yes, thank you, Rosemary. When you asked that question, one very important thing came into my mind. I worked at a CDMO during the COVID pandemic, and what we realized was the disruption of the supply chain, especially supply chain of raw materials, of steel, of equipment pieces, and even of mouthpieces to cover your mouth. For example, when we were working when I was working in a CDMO, we had a laboratory with 20 people working in the same space, and all of a sudden there were not enough face masks in Switzerland anymore. And this is just one example, but generally speaking, what we realized, especially on the manufacturing field, is that before the pandemic, equipment suppliers, they said, okay, you will have this equipment in 18 months or in 14 months. After the COVID pandemic, and now also influenced, unfortunately, by the geopolitical conflicts, they don't promise any timelines anymore. They say, okay, it might take 18 months or 19 months, but it might also take 2 years. We cannot promise a clear timeline. This is one clear impact that we realized at the CMC level. Yeah, thank you.
And then moving on to Alan, your comments?
Yeah, sure. So something maybe different in research, we depend on partnerships and on personal relationships. At GARDP Discovery, we have an external research model, and a lot of that is through personal interactions, and 2024 now is the return to the conference season, which allows, personal relationships to be built again, and I think it's really important, so that to kind of build ourselves back as a community and establishing those networks, which then lead to collaborative projects and working with the best players in the global field. Okay, and I think obviously it was really challenging during COVID times, and now as we see a return to normality for on that level. Yeah.
I'm going to refer to our host because I'm having problems seeing some of the questions online. Victor, have you got some questions for us?
Yes, Rosemary, you should be able to see them if you click the button next to the chat icon with the question mark in there.
Yeah, unfortunately, I haven't seen them. Would you mind reading them out to us?
Sure, I can read one that we've received. So this is for Herbert. So what are some of the ways that new medicines can be made affordably?
Thank you. There are several ways to make new medicines affordable. Obviously, during research and development, you don't have much time to focus on really optimizing the process to make it super profitable. You should be happy enough to bring it to launch, to bring the clinical trials to a successful end. As I mentioned, for example, one way is to increase the batch size. This is one very effective way. Also to simplify, for example, the synthesis process. Or for example, you can also work in manufacturing campaigns. So instead of manufacturing one batch every month, is for example to manufacture 10 batches in the same month and performing the release analytics also the same month. Like this, you develop some very important synergies. Lastly, also one way to reduce the costs is, of course, to outsource to markets in countries that have lower salaries, for example, lower salaries than in Europe or in the US, which we are facing. However, this, on the other downside, has made many governments also realize, especially during the COVID pandemic, which brings me back to the previous topic, that many local governments realized that we were too dependent because we were relying on outsourcing everything to Southeast Asian countries. And during the COVID pandemic, it was clear that we need also maybe to source back some of these manufacturing capabilities and the know-how. So, but to your first question, yes, there are ways to reduce the costs of commercial manufacturing. Certainly outsourcing is one way, but also there are some technical ways to do this by increasing the batch size or making manufacturing processes more efficient and robust. Thank you.
Any other questions?
Okay, one more for Alan. What efforts are put in understanding better resistance itself, e.g. mechanisms, evolution, impact, transmission, and to what extent do you think this knowledge could help designing new efficient antibiotics?
Yeah, I think that's a good question, Victor. I think it's always important, even early in the discovery phase, to know what the future threats will be and to see emergence of resistance as early as possible. We see the WHO reports, for instance, on the priority pathogens, okay, and other reports indicating, the emergence of novel resistant mechanisms and so on, be they target site based, be they through increased efflux or decreased penetration as described during the talks today. So understanding the mechanisms and which agents they affect, and it may be even a brand new antibiotic class, that is addressing a new target may still be susceptible, for instance, to efflux mechanisms where they're not class specific. So I think understanding and screening against such strains early in the process can be really important, not to be falling foul of issues later in the process where it's obviously you've less control on the changing your candidate late in the day. Okay, early in research, you can optimize your chemical class, appropriately to overcome certain resistant mechanisms. Yeah, but very important to understand, to test, and to deal with such issues early.
Okay.
Can I invite any other comments from the panel? Perhaps I would just comment, and I think all three speakers have spoken about the importance of a coordinated team. That doesn't necessarily all happen in-house. How difficult is it to coordinate a team and move these discovery pipelines along when you're working across continents, time zones, and capacities? Are there any
Maybe I can
volunteers?
first. Thanks, Rosemary. Yeah, I think the challenges go beyond time zones and logistics. I think it's such a multifaceted issue. It's not just the scientists or industry partners who's responsible for this pipeline. So we also, Advanced ID also works on antibiotic access issues, for example, and that alone would involve government policymakers in coming up with incentives, how to apply the incentives, not just for the industry, but also coming up with policies in the national insurance level, for example, and also practitioners, the hesitancy and sort of almost apathy in trying to get new antibiotics or effective antibiotics into the market. There's so many different stakeholders involved, and every country has a different legal system, different regulatory approval process, and a lot of challenges. I think the upcoming UN meeting is a great start for political will to handle this issue together. But we also need a lot of local champions too, and they should be incentivized to work on this issue. We need leadership and a clearer direction to move ahead together.
Because we really need to be planning for the next pandemic, whatever it's going to be. And all of these connectivities and interactions are going to be important, irrespective of what the next challenge is going to be, right?
Yeah.
Herbert, would you like to comment?
Yes, thanks, Rosemary. I think it's, well, very important. It's communication, communication, communication. Definitely, with the time zone difference is one important challenge. Also, the cultural or maybe some language barriers is also a challenge that requires mutual understanding and awareness. And of course, you need to record everything you do, you decide, have meetings, write minutes, so there are no misunderstandings. But definitely for an organization like ours, GARDP, it's essential that we have external partners and we have a decentralized way of working. We are working with partners in Asia, but also in the United States. And that requires, of course, a lot of communication. This is something one needs to be aware of. It's communication, communication, communication. That's how I would answer your question.
Alan, one final comment before we wrap up?
Sure, I could agree with my co-presenters there on those points, and maybe, 15, 20 years ago, we had very integrated drug discovery at, major pharmaceutical companies, okay, where everything might be under the same roof in one research center, and now that has totally changed, okay, there's often big pharma still in antibiotic drug discovery. We have, the likes of Roche and Genentech and Merck, GSK, but many of the majors have left and have been, the gaps filled by biotech and other organizations. And this has required a different way of thinking, and even the big players will have large offshoring of their some of their activities, So yeah, definitely, strong links with your partners and communication, as Herbert said there. Yeah.
And I guess that underlines the importance of organizations like GARDP in bringing disparate groups and bringing allowing researchers from across the pipeline to facilitating for them to get together to fill the gaps in the discovery pipeline. I see we're almost out of time, so I'm going to hand back to our host, Victor.
Thank you very much, Rosemary, for moderating today's webinar. And of course, thank you to all the speakers, Alan, Mo, and Herbert, for your presentations. They were all very insightful. So as well as accessing the recording of today's webinars and the resources that we have on Revive, you can also learn more about antimicrobial R&D by joining us for the next Antimicrobial Chemotherapy Conference. This will be held on the 4th and the 5th of February 2025, and it's an online conference organized jointly between GARDP and BSAC, the British Society of Antimicrobial Chemotherapy. So registration is now open and it's totally free of charge, and you can submit an abstract for an oral or a poster presentation before 15th November. So our next Revive webinar will be announced very soon. Please keep an eye on our website as well as our newsletter and our social media channels of GARDP to be the first to hear about this. Thank you very much everyone for joining, and please do join us again in the future and remind your colleagues of these webinars. Please fill in the survey after the webinar to let us know how you found this new interface and also to give us any suggestions for future webinars. Thanks and goodbye.
Thank you very much. Goodbye.
Bye.